Okay, today we'll be discussing cellular respiration. So cellular respiration is the process in which glucose is turned into energy in the form of ATP, which is also known as adenosine triphosphate. This is also called an oxidation reduction process or a redox reaction, when electrons are passed from one molecule to another. When losing electrons, one is oxidized, and when gaining electrons, one is reduced. There are four main stages in cellular respiration in which we will touch base on: glycolysis, pyruvate oxidation, Krebs cycle, and oxidative phosphorylation in that order. So the first process is glycolysis, and that happens in the cytoplasm. At the beginning of glycolysis, a 6-carbon sugar is converted into 2 molecules of pyruvate, which is a 3-carbon organic molecule. ATP is made, and NADH is turned into NAD plus is turned into NADH. And there are 2 main phases. The top part of the image shows the energy requiring phase. In this phase, glucose gets rearranged and it requires 2 molecules of phosphate, turning 2 molecules of ATP into ADP. This leads to an unstable modified sugar, which splits to create 3-carbon compound. The second part of the image is the energy releasing phase. During this phase, each 3-carbon compound turns into a pyruvate molecule, in which results in 2 ATP molecules and 1 NADH molecule. So 2 3-carbon molecules go through this process, resulting in 4 ATPs and 2 NADH molecules from the original 6-carbon glucose. The next step is the pyruvate oxidation. This happens in the matrix. During this phase, you convert pyruvate into acetyl-CoA, which is a 2-carbon compound attached to coenzyme A. A carboxyl group is removed, causing the release of carbon dioxide, and this causes NADH to be reduced into NAD plus to be reduced into NADH. The acetyl group is attached to coenzyme A, which causes acetyl-CoA. The next step is the Krebs or the citric acid cycle. It enters as a 2-carbon compound and turns into a 6-carbon compound that turns back into a 4-carbon compound, which is reused for the next cycle. So in the first step, acetyl-CoA joins with 4-carbon molecule oxaloacetate and releases the CoA group and creates a 6-carbon compound called citrate. This soon turns into a 4-carbon compound, losing carbons as carbon dioxide. This 4-carbon compound releases energy and results in GTP, NADH2, and NADH, and finally turns back into oxaloacetate, which is used again in the next cycle. The last step is oxidative phosphorylation. This is where most of the ATP is generated, utilizing the electron transport chain and chemiosmosis. The electron transport chain is located in the inner membrane of the mitochondria. No, back. Delivery of electrons by NADH and FADH2 turn into FAD plus and FAD, which can be used in the process once again. Electron transfer and proton pumping is where the electrons get transferred through the proteins, getting less and less electromagnetic until you reach oxygen. The byproduct, which is oxygen, combines with 2 hydrogens in order to create water. While the electrons get passed down, the proteins go through conformational shape changes, which allows the H plus ions to pass to the other side. This creates a proton gradient. The last step is chemiosmosis. Chemiosmosis utilizes the proton gradient that was created earlier and allows for H plus ions to go through the protein ATP synthase, moving down the electromagnetic gradient, going from high to low. The H plus ions combine with ADP in the matrix to create ATP. This process yields the most amount of energy, or ATP, with a total of around 30 ATP per molecule of glucose. While glycolysis is an anaerobic respiration, meaning that it requires no oxygen, the other processes are aerobic and need oxygen to be carried out. So in moments in which there is no oxygen, cells go through the process of anaerobic respiration. There are two types: lactic acid fermentation and alcohol fermentation. I guess that's it. Yeah. Thanks for watching.